Good morning and thank you. My name is George Magrath. I'm the CEO of Opus Genetics. We're a company that's based on gene therapy for inherited retinal diseases, and I'm going to be showing you today the clinical data from our lead program. So the company was founded in 2018. We are in the Research Triangle Park, and we are publicly traded under the ticker symbol IRD for inherited retinal diseases. We really have two clinical stage programs, the first of which is Best disease, and the second of which is LCA5. Best disease is a more common inherited retinal degeneration at around 9,000 patients incidence, and we have opened an IND and we're now enrolling patients in the first trial there. Today I'm going to show you the results from our LCA5 program, where we have 6 patients of data in an ultra-rare condition. Now, LCA5 is a disease of childhood where the photoreceptors are missing a protein called lebercilin. Lebercilin is a structural protein involved in the transport of the factors of the visual cycle, and is responsible for the photoreceptors having that elongated shape, or the elongated tail end that people are used to seeing. So in these patients, you end up getting a loss of the photoreceptor kind of tail end, which leads it to not be able to sense light. By restoring lebercilin, we think that at least in animal models, and now what I'll show you in 6 patients, is that by restoring that lebercilin protein, you can restore the function of those photoreceptors. This is a disease of childhood. It really occurs in the first year or 2 of life, so the parents usually notice that the child can't pick up toys, has trouble crawling around, walking around, things like that. Take them into the eye doctor. It's a pretty characteristic exam finding, and then a pretty simple genetic test to identify it. What we've done so far is we've enrolled 6 patients, as I've mentioned. The first 3 we enrolled in the trial were adults. That's patient 01, 03, and 04. These patients now have 18 months of follow-up. On the results of the 3 patients, earlier this year we went to the FDA. We were awarded RMAT status for the results of those 3 adults, and the mandate was to go treat 3 children. And so that's exactly what we've now done, and that's the results that I'll be presenting, and this is the first presentation of these results. So we have 3-month data on patient 05, 06, and 07. You can see that we started with a 34-year-old and we've progressively gone into the teenage years. That's important because what you have to show efficacy is viable photoreceptors. Remember, this is gene augmentation using a standard AAV approach to photoreceptors that are missing a single protein, lebercilin. And so if you have present photoreceptors, you should be able to restore function by restoring that protein. Now, the baseline visual acuity here, you can see this is a logMAR scale. So in a logMAR, 0 would be 20/20 vision, and then when you get up above 2, you're talking about barely formative vision. So if you're above 2, you're talking about seeing blobs and things like that. So these are profoundly visually impaired people. All of them are legally blind. So it's a very safe treatment to date. So all 6 patients have had no treatment-related adverse events. It's been very good so far. There are no serious ocular adverse events and no treatment-related serious adverse events, obviously. When you look at visual acuity, the first thing I'm going to show you is this adult cohort. These are the first 3 patients. And what you're going to see on the left side of the slide is the change from baseline of visual acuity, where the orange line is the study eye, and then the teal line is their control eye. And this is improvement in logMAR visual acuity. So each 0.1 improvement in logMAR is about the equivalent of 5 letters on an eye chart or 1 line on the eye chart. So you can see here that, for example, the average improvement is close to 0.4, which is the equivalent of about 20 letters on the eye chart of improvement. So a pretty dramatic improvement in these patients in visual acuity. And really the most dramatic was patient 03, who went from never having formed vision, a Braille reader his entire life, to being able to see the big E on the eye chart, which was pretty remarkable. On the right side of the slide, you can see the mean intraocular difference, showing that there was a significant improvement compared to their fellow eye. When you look at the pediatric patients, now we're looking at only 90-day data, but what you see is a pretty remarkable improvement as well. And so again, about a 0.4 improvement in logMAR vision, or the equivalent of 20 letters on the vision chart. If you break this down by patient, this is quite interesting. So there are different phenotypes of this disease. So if you look at the data for patient 01, you would say, oh, well, that patient had a less dramatic visual acuity improvement. But she's actually going to be featured on Good Morning America in a week because of how much this has changed her life, because it really affected her peripheral vision. So she had a phenotype of the disease where rods were more affected, and it really led to outstanding gains on other measures that I'll show you in a minute. And so while visual acuity is great, it only measures the central macular function, and it doesn't measure the peripheral retina as well. And so that's why we do multiple endpoints. If you look at FST, this is the first other endpoint that I'll show you. So FST is a test where you really just have the patient, you shine a light at the patient at different luminances and have them respond by clicking a button. And this is a logarithmic scale, and a 2-log improvement is really about the theoretical max for what you could get if you were to, say, improve, all of the retinal function. And so you can see here it's going in the right direction for every patient, with in the adult cohort here on both red and blue. And red and blue are just different wavelengths which are going after different types of cones. In the pediatrics, you see a very nice separation from the study eye, from the treatment eye in the study eye across all 3 patients and across both wavelengths. Now we're getting into data on what we call the MLOMT. This is a multi-luminescent maze. And so Luxturna, the first approved genetic medicine, which I'm sure you guys are all familiar with, was a gene therapy delivered in a very similar manner to what we're doing, that was going after a different mutation, a mutation for RP65. So it was approved in 2017, and it was approved on a maze where they would set up in a conference room, about the size of the stage, really. You would put obstacles, and you would watch the patient, navigate through those obstacles. Well, we've come a long ways since then, and what we're doing now is we do it in virtual reality. So you actually put on the Oculus headset, the Meta headset, you have the 2 joysticks in your hand, and in a virtual reality world, you follow a red arrow, and you tag objects that are presented in that world. So you tag a ceiling fan, you'll tag a table, you'll tag a skateboard, things like that. And so on the y-axis, what you're seeing is the number of objects they were able to identify. So there are 9 objects presented to the patient, and you count the ones that they identify versus the ones that they miss. On the x-axis, you're looking at the brightness level in the headset. So at 0.02, you're like a moonless night, and then at 0.47, you're at like a bright conference room. What we're in right now is probably closer to 0.07, and usually what you're in at home, like with lamps on in a normal kind of house, is the 0.22. So what you can see here is the different colors are different time points. And for example, in patient 01, remember that patient that had more peripheral vision improvement and less central vision improvement, that patient had a dramatic improvement where at baseline, she saw no objects in that eye. However, at every time point afterwards, she saw a significantly improved number of objects at least the 0.07 and above brightness levels. So that's a pretty remarkable improvement for her, and I think that's why she had a really clinically meaningful response too in her daily life. Now, if you look at patient 0103, what you'll notice here is that this patient had the biggest improvement in visual acuity, so so when he stares at something, he could actually see for the first time. However, his peripheral vision didn't improve that much, and so you don't see as big of an improvement on this maze test. So when you think about people's vision, you kind of have to put all of these metrics together to get at what the patient's experiencing. These are the 3 kids, and so what you'll see in these 3 kids is in patient 06, you had a pretty dramatic improvement at all luminances, and in patient 05, you saw some improvement in the 2 higher luminances, and less of an improvement in 07. And again, this goes back to the phenotype of disease. So what we've shown is that there's meaningful improvement in visual acuity of 5 out of the 6 patients. The gains happen as early as a month and really max out around 3 to 6 months. And then multiple endpoints are all kind of going in the right direction. And so this, combined with the patient's experience in this ultra-rare disease, lead us to believe that these patients that we truly might have biological engagement of the drug and really might be affecting these patients' pathology. So where are we now? This study is funded by the FDA Orphan Products Division under a clinical grant there. We are meeting with the FDA currently to talk about what the next steps would be, whether that would be continuing a dose escalation phase 1 or starting to pivot into what would be a potential pivotal trial. And again, with it being 200 patients in the United States, the trial design will be a little bit different than it would be for more common diseases. So that's about where we are. This last page just shows sort of what the patients were saying afterwards, and it's really this is really kind of my favorite slide because every one of the patients absolutely had their life changed by this. These are dramatically visually impaired patients that all had clinically meaningful improvements in their vision. And so it's really great to see, and we're really looking forward to the next steps in this program and also our Best One program. And thank you. That's all we have.